plant and animal cells. This lesson we discuss under 5 subunits. Through this video, we learn about the concept of the cell theory, structure of the cell theory, basic unit of the life, and some experiment related to this lesson. And we finally answers to the task sheet. Okay, shall we start the video? Basic unit of life. When we are talking about the plant and animal body, we should definitely know about the cell. I think you are already know the what is the cell. Before that, we should learn about the history of the cell. When we are learn about the history of the cell, Robert Hooke is a special person because he is an English scientist in 1665. He is the first person who observed a cell by his own microscope, and he observed a cork under his microscope, and he uh, is, discovered the structure like canvas in behave, and that is he named as cells. This is the picture of the Robert Hooke, and you can see the Robert Hooke's microscope, and he observed cell also. Uh, Now we move to the cell theory. When we are talking about the cell theory, we should identify the person who introduced the cell theory. They are Schleiden, Schoven, and Rudolf Schleiden is a German botanist, and Schleiden is a German botanist. Schoven is the German zoologist. This picture shows the pictures of these three persons of Schoven, Schleiden, and Rudolf. Next, we learn about the content of the cell theory introduced by those three people. Content of the cell theory. They propose cell theory are the structural and functional unit of life. All organisms are made up of one or more cells. New cells are formed from pre-existing cells. These are the content of the cell theory. We move to the further learn about these cell theory. Further explain about these content of the cell theory. Cell is the smallest structural unit of the organization of the living body. It means cell is the building unit, like as when we are get the example like house, we can get the any sample of the house. We can see the bricks, like as our body also, every place have a cell organization, because every system, every organ, and every tissues also are made up of the collection of cells. So we can tell the cell is the smallest structural unit or building unit in our body. And the next is we learn the second cell theory. All organisms are made up of one or more cells. In the world, every animals divided into two groups, unicellular and multicellular organism. We learn previous grades. Organisms composed with only one cells are called unicellular organism. They have only one cell, just only one cell. We can get the example like uh, protozoa, bacteria, some fungi, some algae can be taken. You can see that they have very small structure, very simple structure, because they have only one cell. The other kind of the living being in the world are multicellular organism. They have two or more than cells, because they have a complex body structure, not a simple body structure like that unicellular organism. They have very complex structure. In animals, plants, some algae can be taken as a unicellular organisms examples. The other concept is different cell perform different functions. The first theory says the cell is the small structural and functional unit. unit of life. It means every cell did a particular function. We can get the example RBC. RBC means red blood cells. Red blood cell doing the transportation of oxygen. I in that point I give the extra knowledge for that. RBC have a nucleus. And next example is neuron. Neuron is the transmission of impulses. An epithelial cell do the covering the body secretion and absorption. We get just only 3 cells. These cells doing the unique functions to each other. It means we can prove that stands as different cell perform different functions. Uh, uh, we can't see every cell didn't do the common function, did the particular function to uh, in our body. These are the concept of the cell theory. Now we are move to the structure of cell. Before to that structure of the cell, let's study the animal cell and plant cell. First, to study the animal cell, we get some experiment. To do that, we get our cheek cell in our cheek. Now we watch the video how to what is the observation conclusion in that experiment.
Human cheek cells. To do the experiment, we require toothpick, cover slip, methylene blue solution, glycerine, glass slide, distilled water, brush, needle, compound microscope, filter paper. Procedure. Take some distilled water using a dropper and put it in the middle of the glass slide. Take a clean toothpick and gently scrape the inner side of the cheek. Rub the scrap on the glass slide that contains water. Mix the mixture using a needle and spread it. Take methylene blue solution using a dropper and add a few drops to the mixture on the slide. After 2 to 3 minutes, remove excess water and stain using a blotting paper. Take glycerine using a dropper and add a few drops of glycerine to the test mixture. Take a clean cover slip and lower it carefully on the mixture by using a needle. Using a brush and needle, press the cover slip gently to spread the epithelial cells. Remove extra liquid around the cover slip using a blotting paper. View the slide under the compound microscope. Observations. A large number of flat and irregular shaped cells are observed. The cells do not have a cell wall. However, each cell has a thin cell membrane. A deeply stained nucleus is observed in the center of each cell. No prominent vacuole is observed in the cells. Conclusion. As the cells observed do not have a cell wall or a prominent vacuole, the cells of the specimen on the slide are animal cells.
Okay, let's study the plant cell. To do that activity, we need anion epidermal cell. We can get the easily that material in our kitchen. Now we watch the video.
Onion peel. To do the experiment, we require cover slip, glycerine, glass slides, onion, watch glasses, distilled water, saffron in solution, forceps, brush, needle, compound microscope, blotting paper. water. Take some distilled water using a dropper and put it in a watch glass. Take half a piece of onion from the watch glass and remove a leaf from the onion piece. Remove a piece of transparent onion peel with the forceps. Put the epidermis into the watch glass containing water. Take some saffron in solution using a dropper. Put a few drops of saffron in solution into another watch glass. Using a brush, transfer the peel into a watch glass containing saffron in solution. Keep the epidermis for 30 seconds in saffron in solution so the peel can become steamed. Using the brush, take the peel from the saffron in solution and put it into the watch glass containing distilled water. Using a dropper, take some glycerine and put 2 to 3 drops of glycerine in the middle of a dry glass slide. Using the brush, transfer the peel to the slide containing glycerine. Take a cover slip and place it gently on the peel with the help of a needle. Remove the extra glycerine using a blotting paper. View the slide under the compound microscope. Observations. A large number of regularly shaped cells are lying side by side. Each cell has a distinct cell wall. A distinct nucleus is present on the periphery of each cell. Lightly stained cytoplasm is observed in each cell. A large vacuole is present in the center of each cell, surrounded by the cytoplasm. Conclusion.
Okay, let's move to study the typical cell. The cell prepared by including all the kinds of organelles are called the typical cell. What is the meant by organelles? Organelles means the cell have different structure to perform different function are called the organelles. It means we simply take organelles means the part in a cell. So the cell prepared by including all the kinds of organelles are called the typical cell. This is the picture of the typical cell. But remember, we can't never see the typical cell in the world because every cell haven't every organelles. We can get the example RBC. RBC haven't a nucleus, like that every cell haven't any other organelles. So we have to study the typical animal cell and typical plant cell organelles and shall move to the typical animal cell. This is the structure of the typical animal cell. All animals are covered by a plasma membrane or cell membrane. It is a live semi-permeable membrane as well as selective permeable membrane. There is a centralized nucleus in an animal cell. The cytoplasm is a gelatinous material. This is we can name the organelles in the typical animal cell are plasma membrane, and rough endoplasmic reticulum, smooth endoplasmic reticulum, nucleus, nucleolus, nuclear envelope, and ribosomes, mitochondria, and Golgi complex can be taken. Next, move to the typical plant cell. This is the structure of the typical plant cell. The outer covering of the plant cell is the cell wall. It is made up of cellulose. Inner to the cell wall is the plasma membrane or cell membrane. At the center of the plant cell is a large vacuole. Generally, there is no such vacuoles can be seen in animal cells. We can name the typical plant cell. Parts are cell wall, cell membrane, mitochondria, nucleolus, nuclear envelope, nucleolus, chloroplasts, and Golgi bodies, ribosomes, smooth endoplasmic reticulum, rough endoplasmic reticulum, and vacuole cytoplasm can be studied in the our syllabus. Animal cells as well as plant cells possess different organelles. We learn now that perform different functions. I told earlier every part do particular function. Most of the organelles cannot be observed through the light microscope. Therefore, the electron microscope should be used to see the the uh, typical plant cell structure. Now we move to the differences of animals and plant cell. There are some similarities also can be seen in animal and plant cell. When you get the example, plasma membrane having animal cell and plant cell. Like that, there are some differences between animal and plant cells. Cell wall is absent in animal cell, but cell wall is present in plant cell. Large content of it contains cytoplasm in animal cell. Cytoplasm is pushed towards periphery in plant cell. A large vacuole cannot be seen in animal cell, but a large vacuole can be seen in plant cell. But animal cells, they are in rarely can be seen some few small vacuoles present, and chloroplast never seen. Animal cell, chloroplast present in the plant cell. These are the differences between animal and plant cell. This is the theory of the today video. We move the today task sheet. These are the questions I give to you. Answer to it, and can you send you can send in put in the comment box. I hope you are really enjoy my lesson. So don't forget to subscribe my channel, like and share. If you have any issues related to this lesson, please comment into the comment box.